This is the first protocol that allows human breast tissue to be maintained alive ex vivo for prolonged periods of time, directly allowing the study of interactions between human breast tissue and human breast cancer. The main advantage of this technique is that it allows macroscopic pieces of human breast tissue, including breast adipocytes, immune cells, vascular structures, ductal structures, and extracellular matrix, to be kept alive ex vivo. This technique has major implications for several areas of breast cancer research, including personalized medicine, pharmaceutical development, and the pathophysiology of tumor initiation and slower breast cancer processes, such as fibrosis and extracellular matrix remodeling. Demonstrating the procedure are Katherine Hebert, a Tulane graduate student, and Rakesh Gurrala, a Tulane medical student from my lab.
To begin, melt the prepared gelatin solution in a 37°C water bath. Use a 5 or 10 milliliter serological pipette to dispense 2.5 milliliters of this solution onto each plunger for wells of a 6-well plate. Once the gelatin has solidified, move the P-NIPAM-coated ASC plates to the biosafety cabinet. Gently place the plungers into the wells of these plates so that the gelatin contacts the ASCs. Place a metal washer on the plunger to weigh it down so that the gelatin is in direct contact with the ASC sheet for 30 minutes at room temperature.